Welcome. Okay, this is Lecture 5, and this focuses on the chapters 10 and 11. Excuse me. Now, identifying and classifying microorganisms as well as the diversity of prokaryotes. Now, there's going to be a little caveat here. Please follow the lecture notes very carefully. Reason? I'm clipping out some of the more, esoteric parts, particularly of Chapter 11. Now, for those of you that keep in mind, remember, this is a microbiology course for healthcare providers. It's not a clinical microbiology course in the sense that you have to diagnose everything, and it's neither a general microbiology course where you have to understand every microbiological organism that's out there on the planet, including those that eat dirt, eat mining waste, and work in septic tanks. No. So, some material I'm going to remove, and that you will not be tested on, so you don't have to worry, but I would follow closely with the notes, okay? Now, first off, we're going to start off with Chapter 10, Identifying and Classifying Microorganisms. We are going to go through the principles of taxonomy, so basically the science that studies organisms in order to arrange them into groups. I encourage you, of course, to review the key terms and the glimpse of history. Keep in mind that this particular section describes the evolution of microbial identification techniques. So let's go ahead. Now, based on taxonomy, based on these strategies, we come into identification, classification, and nomenclature. Identification is characterizing the organism. What is it? Classification, arranging the organism into similar or related groups, and it allows for easy study and identification. How are these organisms related? Finally, nomenclature, a system of arranged names to the organisms. So what do we call it? So when you look at the strategies to identify, we start off with structural, but there's limits to that, right? You know, it's round, it's rod-shaped, it's spiral. Yeah, that's great, but there's lots of round, rod-shaped spiral. We start biochemical. Okay, gram staining, capsule staining, acid-fast. Okay, there's lots of gram-negative, gram-positive, so we got to get into further biochemical tests, which you've been touched upon earlier, okay, those that will break down gelatin, those that will make certain acids, those that will make certain gases, those that metabolize selective sugars. Okay, finally we get to genetic approaches, and it's the genetic approaches that really have been able to help us to expand and understand strains and subspecies and things like that. And so we understand things a lot better. But all of them taken together allow for us to understand the species, the genus, etc. And this is extremely useful because as much as you might think that this gets a little bit boring, it's critical to understand. How do we identify what particular treatment is useful for the patient? How do we sterilize food or medical products, etc.? In strategies for classifying, this is where we're going to come down to the structure, the phylogeny, which is the evolutionary relatedness of organisms, and it allows for classification of organisms based on how and when they were developed. Now, there's a challenge here. We got a problem because, you know, it's easy to sit there and say, okay, first we had fish, then we had amphibians, and somehow we ended up with reptiles, and off of reptiles came birds, and then some reptiles formed into mammals. They had a big comet hit the planet. Most of the big dinosaurs died out. Boo-hoo. Okay, you see where I'm going with this. You probably experienced a little bit about this, okay? Classification of organisms based on how and when they develop. Problem. The definition of a species of plants or animals, that can get really straightforward because plants and animals don't breed with each other. And we define species as usually organisms that cannot breed together and yield a fertile offspring. That's at the animal stage. You take a horse and a donkey, and what do you get? A mule. But a mule is sterile. Okay? So it doesn't create, and that's basically demonstrating what we call species boundaries, okay, between horses and donkeys. But it can't be applied to bacteria. Bacteria reproduce by binary fission. They don't mate with another related opposite sex organism. And also we get all this stuff that you've been dealing with the past couple of weeks, particularly bacterial genetics, etc. So you start having situations where evolutionary relatedness can't always be made merely by structure and biochemistry. Biochemistry would be considered phenotype. It may require the genotype, genetic analysis, but then we got another little problem here because, well, bacteria exchange genetic loops of DNA. Remember the plasmids? Okay. Now we've come up with taxonomic hierarchies. This is the base of hierarchical classification, and it's starting with species. By the way, on page 246, know those definitions of the terms. Species, genus, family, order, class, phylum, which is division, kingdom, domain, da-da. Now, you can see them here in table 10.1. Domain, it's bacteria. Phylum, proteobacteria. Class, which is a subgrouping now, gamma proteobacteria. Order, Enterobacteriales. Family, Enterobacteriaceae. Finally, genus, Escherichia. I remember that one. And then the species, coli. So, there's your example. And if you go back to the history part, glimpse of history, what happened was this. We had up in the 50s and 60s and early 70s a wonderful system by Whitaker who basically divided the 5 kingdoms. Bacteria, plants, fungi, animals, protozoan. But the system failed to consider archaea and bacteria as separate. Then we came back to Woese, and W-O-E-S-E, Woese, used the analysis of the ribosomal RNA, the 16S unit. And he basically creates 3 domains. Archaea, bacteria, and eukarya, or the eukaryotes. In other words, everybody else, everybody that's got a membrane-bound nucleus. And you can see that that has some considerations. Now, there is a manual that kind of defines things. Oh, yeah, and by the way, table 10.2, really nice. Gives you some basics about the comparison. But if you take a look on table 10.3, this is where we get into what's called the Bergy Manual of Systematic Bacteriology. Bergy's has got several different big, thick, they're almost like mega thick like Bibles, compendiums. And they're reference texts used to classify prokaryotes. Contains information on the organisms, the ecology, the methods of enrichment, the methods of culture, isolation, as well as the maintenance and preservation of the organisms. In other words, they might talk about, we found this particular bacteria growing underneath the rotting wood in a sequoia forest in spring, and we found that we can isolate it and what particular cultures it goes with, etc., etc., etc. Okay? That's just so that you're familiar with these things. And remember that we did say this at the very beginning of earlier in the course, that only 1% of all of the microorganisms have been successfully isolated and cultured on their own. Some of them are too tightly interactive with other organisms, and we have not been able to develop ways to separate them and culture them alone. Now, when we get to nomenclature, we get to naming rules. This is set by the International Code for Nomenclature of Bacteria, and there are the rules. Rules are pretty straightforward, actually. Bacterial names, if you want to talk about this, let's go back here. Bacterial names may originate from any language, but they must be given a Latin suffix, so a Latin ending. Name may reflect characteristics of the organism or used to honor a prominent researcher. And this goes on, by the way, also with the formation of genus and species names for a lot of the new organisms that are found on the planet. Somebody comes up and they said, you know, they want to name it after their favorite rock star or something else like that. Great. Great. Now, during periods of name change revisions, former names of organisms may be included in parentheses. So if you note in my notes, I say Lactococcus used to be referred to as a Streptococcus lactis. It's now known more officially as Lactococcus lactis. Okay? So, moving forward, we need to talk about, for a few minutes, the phenotypic characteristics of identification. Now, phenotype, if you have to understand, you've never really dealt with this. This is observable characteristics, not genotype, which is the genetic makeup. Phenotype includes their morphology that you can see under a microscope. How are you culturing them? What are their biochemical capabilities that you would see in biochemical tests? Perhaps you use serological testing, okay? Certain antibodies will react to them. Maybe they make certain proteins that you'll be able to get generated on a certain type of analysis tool, such as MALDI-TOF mass spectrometry. But we'll go on to that. So let's start off first with, and you got to keep in mind that phenotype has certain limitations even in and of itself, because you don't know what other genes are in there, and yet the phenotypic arrangement and the phenotypic tests have been around for, in some cases, going back over 100 years. They're not as reliable as genetic, but then again, the genetic tools are not always available, and you have to go with what you've got. Now, as I said also, these are favored in simple discernment, but they don't require sophisticated instruments, and they can be done easily anywhere in the world. So, microscopic morphology, the methods would include, obviously, size and shape, can include grouping behavior when observable, Gram stain or differential stains used mostly in clinical settings, special stains useful where Gram stains fail, such as acid fast from Mycobacterium, endospore stains for detection of endospores. If you go to the next table, which would be Table 5, and that's some of the important biochemical tests. There we are. These are extremely powerful techniques, but they've got to be coordinated. They've got to be used in a coordinated fashion, especially in a clinical setting. And a lot of times you're going to get into what's called the dichotomous key. So it's sort of, put it very basically, is dichotomous key is basically saying, okay, for example, look at catalase. Does it basically, the test, does it detect the activity of catalase? So does the organism have catalase? Yes, no. If it's yes, it goes in one different direction of tests. If it's no, it goes in another direction. So the methods that we've talked about for metabolic differences include things like culture characteristics. So the atmosphere, aerobic, anaerobic. Culture medium, basic versus selective. Use of differential culture medium. The medium pH, the colony appearance. Do the colonies have a color? Do they have a particular colony shape? Do they have an appearance? What is their carbon source for metabolism? So remember now, not all of them will use glucose. They might use lactose or fructose or galactose or something else. Okay, got to keep that in mind. Biochemical tests, these are useful to determine enzymatic processes, and if used in a series of various tests, can help differentiate various species from each other from different various strains. Now, in your lab section, you're going to have a couple of those tests from time to time. What I do is just get you introduced to them. I don't demand that you know them to the level of a clinical microbiologist. And if you want to do that, that's fun, but you know what? There's a lot of other fun things, just so that you be very familiarized with them. Excuse me. So that's why I'm telling you, you don't have to memorize, well, what's the methyl red, what's oxidase, what's the phenylalanine deaminase, what's the sugar fermentation. No. But understand that these are just useful tests to say whether yes or no do these have certain characteristics, and that will help you to determine the identity of the organism. Now, some of these various tests determine metabolism as well as the thriving in various environments. Some of them will thrive in acidic environments, basic environments, salty environments, very high osmolarity situations where basically they have to be osmotolerant, okay? And yet we've also found that because of that, we can get certain detections. The breath test, which is used to detect carbon dioxide produced by the metabolic activity of Helicobacter pylori. And I mentioned about the Dichotomous Key. And if you go back to, here it is. There's a Dichotomous Key right there. Now, this is just a flow of tests. You start with a gram stain. So is it gram positive? Then you go down that particular route on the left. If it's gram negative, go on the particular route down the right. Next test, take an oxidase test or take a catalase test. And if it's plus or minus, you're able to make some determinations as you continue further down. Each test gives you more information, whether it's a yes or no, positive or negative. Okay? And they help you to identify the organism in question. Now, there are some commercial test kits that combine biochemical tests basically to reduce the identification time and improve the accuracy of the identification of the organism. Okay? Now, a lot of these are all put together in nice little kits. Here's another example of a dichotomous key, and you can see how that's moving downward. Here is a test. Now, at the same time, you have the same culture. This is an API test strip. And you can do all sorts of tests at the same time. That means that you cut down on the time it takes from when you've taken the sample from the patient, and you can get an identity of all the different biochemical tests all in that one strip for that particular organism. And there are a variety of other tests out there that are similar to that. As you can see here, this is an API test on the left. The other one, and I've done this in my classes, is an entra tube. And the neat thing about that is basically you take a small sample from the colony from the plate. You have a metal rod that's in the center of that, and you pull the cultures up through each one of those individual chambers so it inoculates each of the chambers. You incubate it, and within 24 hours, you've got 15 different tests, aerobic, anaerobic, all the biochemical tests that you saw earlier on that chart. And you can tell all sorts of neat information, positive or negative. And what they do now is they provide you with a little book, and it looks almost like an old TV guidebook. It's got lots of little thin pages. But when you add up all of the information from each one of those tests, you can come clearly with an identification of what that organism is. Now, moving on, there are other types of tests, and I'll just go through some of them. Serology, which basically uses the serum antibodies to measure and detect antigens. Remember, antigens could be fatty and sugar mixtures, or proteins with sugars, or glycoproteins on the surface of the organism that help to identify what they are. Not to mention the presence of pili, cell wall, flagella, and other arrangements in the glycocalyx. There are other analyses too. You can take the organism, run it through a particular test for fatty acid analysis. Bacteria differ in their type and quantity of specific fatty acids in the cell membranes. After you culture the organism of interest, the fatty acids are treated with sodium hydroxide to convert them to fatty acid methyl esters, and this is a FAME, and then they're analyzed using gas chromatography, which creates a unique pattern for each organism. Now, the malty talk that you see up here, and that's what it is, and you see that on page 264. And I think I also tried to give you at least one video, one YouTube video on this. Basically, you've got a bacterial colony, and it's mixed in solution to convert the entire colony into kind of gaseous state via a laser. Okay? This is matrix-assisted laser desorption, ionization, time-of-flight mass spectroscopy, which is a mouthful. Basically, the principle is you take the colony, you briefly vaporize it with a laser. The gaseous particles become ionized, and they're separated out by a chromatographic column. Now, the chromatographic column separates out those molecules that move fast, those that move slow. Then you analyze the set of peaks of those particles that come out over a period of time, which is not very long. As you can see on that rightmost chart, it creates a unique fingerprint to identify the organism of interest. And that is a very, very powerful tool. Now, we move from there to the genetic approach. As we said, you get a much more accurate identification as well as determination of unique strains. This is the key point. We are going to constantly be faced with the development of new strains. They're going to pick up a new plasmid, they're going to have some sort of piece of DNA they're going to grab, and suddenly you've got a strain of bacteria that's resistant to another antibiotic, produces another exotoxin, has an endotoxin, has another enzyme, and so you have to be ready to say, this is a different strain of E. coli than they've ever developed before. That's why we have to shift the antibiotic use, or we have to shift the treatment. Okay? You also have to remember that some organisms can mutate. They may fail to express the gene product during testing, and that'll lead to a false negative, but the DNA information is still there, and that's where the genotypic characteristics, those tests are very, very valuable. First one we've got is nucleic acid probes. This is using probes labeled with fluorescent or radioactive labels. Most of the day now it's fluorescent. Okay? The radioactive stuff, people wanted to get out of the lab as quickly as possible. Now, these probes are going to be specific for the gene of interest for the organism. And it also uses FISH, which we've talked about, that in situ analysis, so that you can do this in an environmental or clinical sample. In some cases, you could do the tests almost right at the site of treating the patient. Of course, we have nucleic acid amplification tests, and that's where we get into things like PCR. And we talk about PCR, and I refer you notice that it's not, excuse me, it is the PCR that you saw earlier, okay? And keep in mind that with PCR, what can happen is it's very sensitive and it can detect sample organisms in food, water, body fluids, and soil. Now, if you think about it, remember the discussion I had before about PCR, polymerase chain reaction, where you can make a lot more copies of that needle compared to the haystack. Then what you do is you add a nucleotide probe, and that probe is just going to completely screen because there's so many copies of that particular sequence of DNA that you're looking for. Now, the next area comes up to an interesting part that we talked about earlier with Woolsey, and that is sequencing ribosomal RNA genes. Keep in mind, ribosomal RNA is sort of the framework, the structure within the ribosome, proteins attached to them. And that's why you see the 16S ribosomal RNA plus 21 polypeptide chains, so 21 little proteins attached to that 30S. But it's the 16S that's where we're very interested in. That 16S is smaller, and the gene sequence is highly conserved, so therefore mutations cannot easily occur as they will interfere with the ribosomal function. The limited changes in gene sequences that do occur have become a means to identify and classify the bacteria of interest. That's where Woolsey was getting into things, and that's why it became so incredible. Okay, is it bacteria? What type of bacteria? Is it archaea? What type of archaea? The focus shifts from ribosomal RNA to the ribosomal RNA genes on the DNA, and this is called ribosomal DNA. Ribosomal DNA is much more stable to handle in the lab. So once you have a database of 16S sequences that have been assembled, and identification and classification of subsequent organisms can occur. And I hope that makes it clear there. Now, we're going to get into strain differences. Now, you have to understand something, that strain differentiation is necessary because some strains cause disease and others don't. And it's the virulence factors that may cause these unique situations to pop up and become very, very difficult. When I say E. coli, some people would think of, oh, well, that's stuff that's found in the gut. Oh, well, that's something I played with in my lab. It was totally harmless. Whereas others would be sitting there going, oh, well, that's what killed my little brother because he had an improperly cooked hamburger at Jack in the Box, and the beef had this very nasty strain of E. coli. Yeah. Now, the strain differentiation will also let us know if the strains are harmless or if they started to pick up new virulence factors. So detecting the strain is very important, especially when you're tracing the source of an outbreak. And I refer you over to the Perspective 10.1 on page 256 to look over that information there. I'm just thinking there for a second there. Different tools are used to distinguish various strains. First, of course, we have is the biochemical typing. We have what's called a biovar, or biotype. This is a strain that has a characteristic biochemical pattern that is different from other strains. Various biochemical tests can distinguish different strains, some due to the strain's unique production of basically unique proteins, enzymes, or their lack thereof. The other issue is serological typing or serological testing. That's when we get into what we call a serovar, or serotype. This is a strain that has a characteristic antigenic structure that differs from all the other strains. Remember, basically, a lot of these strains, they may differ, and some of the proteins that they differ, or the polysaccharides, are going to be present not merely inside the bacteria, but on the external surfaces. And that's going to give a different serological result when you do the tests. Okay? Another type is, and this is the most powerful one, genomic typing. And what you see there, and it's used to distinguish different strains based on genetic differences. If we go, we see that we have bacteria here, and they would have, obviously, capsule, a cell wall, a flagella, and each one, these are H, K, and O antigens, so they would be able to be detected. But here's something even more special. You can, because of genetic differences, identify, using, of course, restriction enzymes to cut up the pieces of DNA, and you can identify the different strains. Now notice this is strain A, B, and C, but because they have even point mutations, single base mutations, that is going to alter what the restriction enzymes are going to cut into fragments. And what you can do is make easy identification of these different strains. All right? Rflp, restriction fragment link polymorphisms, as I said, use restriction enzymes to cut the bacteria genome up, and so you can use pulse field gel electrophoresis. Basically, it's a modified gel electrophoresis. You're going to use the Rflp. You're going to make infrequent cuts, so you're going to have some large pieces, small pieces, and to separate out some of the larger pieces, you have not a continuous electric field, but a pulse field that goes back and forth, and it allows you to really separate out larger pieces, so you can get a good identification on this. Now, there is a modified southern blot method, and this is involved with what they call ribotyping. This is used to analyze the ribosomal RNA genes. Okay? The bacteria have several different ribosomal RNA genes. The probe is used to identify and differentiate between the ribosomal RNA genes of different strains. If you notice at the very beginning of the text, they had some information there about PulseNet. And PulseNet is really an early warning system for outbreaks of foodborne diseases. So you see, if you go to the beginning of the chapter, which I would turn you back to page 255, you have somebody there that's demonstrating PulseNet, and they have a pulse field gel electrophoresis image on the computer. Here's the importance about PulseNet. PulseNet basically is a national network of public labs that perform DNA, basically fingerprinting on the bacteria that may be foodborne. So the network identifies and labels each fingerprint pattern, permits rapid comparison of these patterns through an electronic database at the Centers for Disease Control and Prevention, CDC. See, basically, everything gets forwarded down to CDC, and you would get a back feed where they would sit there and say, we've seen this before, we've never seen this before, this is serious, it's spreading throughout the country, everyone should be alert for it. Okay? The fingerprint via this pulse field gel electrophoresis can distinguish strains of an organism such as E. coli, Escherichia coli, Salmonella, Shigella, or Listeria. But this is at the DNA level, guys. And this is why it's so incredible. So when you hear a news announcement about, oh, you know, your favorite taco place, or, and you notice I'm not going to use any names right here, but your favorite hamburger place or something like that, and there's an outbreak, they, in many cases, the field epidemiologists and the people at PulseNet and the workers at CDC can basically pull together a picture relatively quickly of where this is. Is it local? Is it spreading? What is causing it to be spread? When did they start seeing the outbreak occur? How to treat it? And they may, if necessary, also turn around and say, look, there's a certain packaged food that has to be recalled. That's it. End of discussion. Because it may be contaminated with A, B, C, or D bacteria. Okay, moving from there, we have also another type of analysis called phage typing. Now, you have to keep in mind, there's strains of bacteria. They differ in their susceptibility to various phages. A phage is a bacterial virus. Okay, we're going to be getting into that in a couple of chapters when we start talking about viruses. So what they do is they will culture the strain of bacteria, and it's plated, and they'll add various phages to it. Now, the different phages will respond. They will either react and start to destroy the bacteria because they've got to infect the bacteria to take over, hijack the cellular machinery inside the bacteria to replicate new viruses. And so what happens is you start getting these clear spots on the bacteria culture lawn, as it's called. It looks like lysis. In other words, the cells are being destroyed, but that's really what's happening. Once the viruses start replicating, they will then spill out after they have broken through one cell. The new viruses will infect other cells, et cetera, et cetera, et cetera. And so you'll get these patches where there are the certain types of phages correspond with the bacteria. That's a way to identify this. Now, unfortunately, the use of this technique has been mostly replaced by genetic methods, but it was a very useful method at the time. And then, of course, we've got the antibiograms. And this is antibiotic susceptibility patterns. Now, if you notice there, you've got your Kirby-Bauer method of antibiotic susceptibility. The little paper discs that have been saturated with different types of antibiotics. And so they label them. They also may put pretty colors on them. But basically, you can tell what that lawn of bacteria, that's the spreading out of your particular suspect bacteria, is. You can tell whether it's susceptible. There will be a large zone of inhibition around it because the bacteria cannot function in the presence of that antibiotic. Or in some cases, you've got nothing. Such as if you look on the right side, down where there's sort of a magenta disc, there's no zone of inhibition. That means that the bacteria are not inhibited in any way by the presence of that particular antibiotic. Okay? Now, if you do this, you can get a lot of information on the susceptibility of those strains to antibiotics, and also the pattern can also indicate different information that you can extrapolate into an identification of the strain. Again, use of this technique has mostly been replaced by genetic methods. Now, we're moving forward, and I do encourage you to review the case presentation 10.1 and the microassessment. Now, what are some of the challenges to classification? Excuse me. Basically, we don't know what the first primordial organisms on the planet were. Fossilized older species cannot provide much information, or any information, as a matter of fact, because usually when we talk about the evolution or development of various species, you got to have some sort of fossil. And bacterial species evolution is kind of not really very strong because they don't leave skeletons, they don't leave fossils, and you're really talking about what can you tell at the DNA level? Not much, there's nothing there. There are some evolutionary chronometers out there. Here's the deal. A nucleic acid, DNA or ribosomal RNA, can be used to measure the time elapsed since two organisms diverged from a common ancestor. Okay, so if you take a look up here, you've got a phylogenetic tree. So how do you tell over a period of time how related or unrelated these particular species are? For example, if you look where the yellow is, where it says Flexibacteria, Cytophagia, Flavobacterium, Bacteroides fragilis, you can see where some of them had a common ancestor and they bridged, they basically differed because they started having some mutations, etc. But really, can you tell what's at the very far left where there is anything? Not really. You can tell to a point what certain species are related. Okay? You can tell how they're related. You can base it on some cases their DNA, their RNA, whether they have high C to G, which means guanine to cytosine, or whether they have low cytosine to guanosine in their DNA. Okay? This gives us this concept that we talk about for a phylogenetic tree. Okay? But here we have the problem. When we talk about horizontal, that's lateral gene transfer, gene exchange via conjugation, etc., this can further obscure the development of many bacterial species and even the families. Therefore, the result is thought to be more an evolutionary shrub of life, and that's what you see on 1015. You see here, where did the mitochondria come involved? Where did proteobacteria come? Well, could they have also gotten influenced somewhat by some of the cyanobacteria, which passed over and becomes kind of absolutely confusing? It's got to also be remembered that appearance, phenotype, may not always reflect the genotype of species, since some genes develop, mutate, and are lost. Remember, sometimes, as I mentioned before about some of the processes of, frame shift and point mutation and things like that, you lose what was there, and it's changed. And over time, these species may survive, but you've lost a lot of vital information. That's like tomorrow morning waking up and finding out that, think about it, all jazz music disappeared overnight. So we have no influence and no understanding of how certain music was influenced, like rock and rap and all the other types. Some of them will say they were influenced by these jazz performers, etc. Well, what is jazz? We have no evidence of jazz. Or another way to say it is how sports have developed. And they start turning around and say, well, we got influenced by this or that. Well, the record's gone from that. And so this is why it gets to be very, very confusing when we try to have an understanding of classification of bacteria, okay? And remember that not only will the genes disappear, but also those gene products that might yield cell structure, enzymes, metabolites, etc. Classification to determine the relatedness of various organisms uses the following strategies, but the genetic methods have become most valuable and most interesting. What I'm going to say here is this. We can try to do some analysis, and we can talk about some of the techniques, but there's only just so much you can get out of them. But let's talk about some of the genotypic characteristics. Certainly, they're more favored as a means of analysis. The more genes an organism has in common with another organism, the more likely they are related, and the more closely they are related to each other. There are three major methods of genomic analysis for relatedness. One, and we're going to talk about these, it's the DNA base ratio. In other words, the cytosine to guanine content. Now you're going to see that in a second. Since G binds with C, and this is with 3 hydrogen bonds. Remember, C's and G's, 3 hydrogen bonds, A's and T, 2 hydrogen bonds. The more CG pairs existing in the DNA, the higher the temperature it would take to melt the bacteria. In comparing two species of bacteria, the closer the melting temperature, which would be a reflection of the CG ratio of the DNA, the closer the melting temperature of the DNA from each species, the more related the two species are believed to be. Other means to analyze CG content exist, but it's thought that if the two species differ in C to G content by more than a few percent, then they are unlikely to be related. And here's an example of that. Okay? Remember, C to G, think about it this way if you're having a difficulty. If C's and G's have 3 hydrogen bonds, it takes more energy to cause them to separate than A's with T's. The higher the amount of C's and G's that exist in the DNA of that bacteria, the higher the temperature. The more related these two species are, if their temperature is very close to each other, because their sequences would have comparable C to G ratios, okay? Now, another amount is DNA hybridization. The determination of the relatedness of 2 species of bacteria can be done using a single-stranded DNA from one organism as a probe, and you basically anneal or attach it to the single-stranded DNA of the other organism. Now, if the strains anneal with a high amount, the species are closely related. If not, then they're less likely to be related. Think about it this way. If you took one strand, not both strands, but one strand of E. coli, pop it up against one strand of Shigella, they're closely related, and they should be therefore grouped in the same species. Of course, then we go back to the other approach, which was ribosomal RNA analysis. Analyzing the sequences of rRNA, Carl Woese found signature sequences, characteristic sequences in the ribosomal RNA genes that can be used to classify and identify organisms. It is the ribosomal DNA, though. These are the genes that on the DNA code for ribosomal RNA, okay, that are analyzed. Conserved sequences are limited to changes, and some changes, mutations, are fatal. See, here's part of the point. The more changes you have, if those changes cause real problems with the function of ribosomal RNA, you die. And therefore, you're going to be as conserved as limiting on mutations as possible. Those species that are close enough, okay, they're going to do well. If you have any changes and you survive from that, that may reflect evolution. And yet, identification sequences might be still possible. Now, you need to be aware of this. We've been talking about prokaryotes, prokaryotes, prokaryotes. You have bacteria, and yes, you can apply this also for archaea. But you can also do this process with eukaryotes, except what you'd be using is the smaller part of the ribosomal subunits for eukaryotes, which is the 18S ribosomal RNA sequence. The process is better than DNA hybridization, as it is far more quantitative, which we like, quantitative, as opposed to qualitative. Qualitative, you might say, yes, no. Quantitative is 1, 2, 3, 4, 5. Gives you that straight number. Also, the development of the phylogenetic tree. You've seen this in 1014, and if you go back to it, okay, we had that wonderful big long list, and believe me, they are bigger lists than just this. And this is due to the sensitivity of the 16S RNA analysis. The genetic relationships can be analyzed and mapped out. You get a phylogenetic tree. Now, here's a couple of points to help you to understand these things. The phylogenetic tree is really a type of diagram, and it tells you the evolutionary heritage, the relatedness of various organisms. Individual species are represented by nodes. Okay? You have an internal node, which is a branch point, which represents an ancestor to today's organism, an external node, which represents species that still exist. The species name are included. So if you look over, say here, Rickettsia rickettsii, that's a known species. What's down here? What's over here? What's down here? What's down here? Nobody knows. A liner branch depicts an evolutionary relatedness of the two species. Okay? So this branch here says Rickettsia rickettsii and Caulobacter crescenensis were related in some way. Okay? Deep branching, this is where you have ancient organisms that branch at early points of evolutionary history. Now, deep branching might be these branches you see on the far left, but nobody really knows what's the name of this particular organism or this one back here, etc. Okay? Let's just go over a couple of other points with phenotypic characteristics, using a comparison of an array, not a few traits that are similar. A profile of relatedness can be developed. And these traits include biochemical, structural, and cultural traits. So in other words, we move back from genetics for a second. We say, look, if we apply the same phylogenetic tree, but instead what we do is we use basically phenotypic characteristics. Yeah, we can get a lot of information and perhaps get an insight of relatedness. And the more related the organisms must be, because they have more in common, the more closely related they are. But there are limits, and I have to stress this. We've been thinking that life came around about 3.8 billion years ago in history. And there was nobody around with a video camera or genetic analysis. There was no way to take a picture and say, this is what the bacteria were. These are the first primordial bacteria. So we are striving to do a lot, but we have to recognize our limitations in our analysis. Okay? Now, I hope you got all of that, and I wasn't trying to be confusing there. I do encourage you to take the time to review the microassessment, the future opportunities, and the chapter summary for Chapter 10. We are now going to move into Chapter 11, and I do encourage you to be very astute here, because this is where I'm going to chop out pieces of it where it doesn't have as much relevance to healthcare providers. Of course, who knows, maybe in 10 or 20 or 50 years, I'm going to be eating my words, but that's okay. Keep in mind that, again, you have to always review the key terms on page 275, and also review the interesting area of the work that was done by Cornelius Van Niel. All right, and that's in the Glimpse of History. So when we examine the diversity of organisms, we have two approaches. Basically, we have the metabolic diversity, and that is what is the energy source and what is the source for carbon. Keep that in mind. Energy source, source of carbon. The ecophysiology is what environment does the organism exist in nature. This is especially important for medical issues. This is where you're probably going to go, oh yeah, I remember that one. Oh yeah, I remember that one. Don't worry about that part. But you might also have an appreciation for metabolic diversity when you start understanding that basically some of these principles were why we discerned, do we grow this particular organism with or without oxygen? Do we note that it's going to ferment as opposed to get lots of energy from each molecule of glucose, lots of ATPs? Does it need light? Does it not need light? Does it need sulfur? Does it not need sulfur? Where does all this come from? This comes from basically early Earth was anoxic, meaning lacking totally oxygen. And what happens is, in studying the metabolism, this provides us information, as I said, of where and how the organism obtains energy, the carbon source. It plays a role in determining whether the organism it would be a pathogen or a saprophyte. Now, a saprophyte, you need to understand, is an organism that takes in nutrients from dead and decaying matter. When we get into fungi, that is going to be a big topic. Okay, how does all the tons of organic matter, leaves and trees and skin cells, everything else that breaks off from us, or etc., how does it all get recycled back to nutrients that other organisms can use? Also, on determination, we can determine if the organism is going to be an extremophile, living in extremely salty or low pH or high pH or hot or high pressure. We also have to understand all of the terms that you see there. Energy source, terminal electron acceptor, phototroph, chemotroph, chemoorganotroph. That's a mouthful. Basically, where is it getting its organic carbon from? Okay? Where is it getting its energy from? Chemicals. Chemolithotrope is even more interesting because what it's doing is getting its chemicals and its energy from chemicals deep in the earth, such as the organisms that live in the deep volcanic vents at the bottom of the ocean. There you have very high temperatures. You have just basically these, what they call smokers, where they're kicking out this blackened stuff that really is rich in hydrogen sulfide, and these organisms are all living there. They've never seen sunlight. They don't need sunlight. They don't need light energy to run their processes, their chemical processes. We have fermentation, anaerobic respiration, and aerobic respiration. So if you're not clear on these, take the time to review them. Now, Anaerobic chemotrophs. Okay, and we see some right there. Yep. These are due to anoxic, devoid of oxygen environment of ancient Earth. It's believed to be the first bacterial organisms. They're members of archaea. They've obtained their energy from hydrogen. They use carbon dioxide as a terminal electron acceptor and produce methane gas. And some of those include Methanococcus and Methanospirillum. And if you take a look on table 11.1, you've got all these different characteristics. I encourage you just kind of review them. Do I got to memorize them all? No. Follow my notes. But you should have some understanding basically. I'm not going to sit there and have you memorize some very unique ones, although the ones involving disease you're going to keep coming back time and time again. The anaerobic chemotrophs perform anaerobic respiration. They oxidize organic compounds for a carbon source and energy. Their terminal electron acceptors use a sulfur-sulfur compounds. They are gram-negative. And so desulfo-vibro is one of them. Now, we go to the anaerobic chemoorganotrophs. They perform fermentation. They use organic compounds for energy, but undergo fermentation. So therefore, they're going to release either acids or gases as metabolic waste. The three genus of note. Now, note in correction in my notes. I tell you to go to page 271, figures 11.2 and 11.3. These are going to be ones for the lactic acid, not the Clostridium. Clostridium do endospores. They ferment different compounds, so you need to be aware of them. Clostridium tetani, which causes tetanus. Clostridium botulinum, which causes botulism. Clostridium perfringens, which is gas gangrene. The lactic acid bacteria that you see include Streptococcus, Enterococcus, Lactococcus, Lactobacillus, leuconostoc, which is and all of these are obligate fermenters, meaning they can grow in aerobic environments, but they end up producing lactic acid. They also lack the enzyme catalase. Proprinium bacteria are gram-positive, pleomorphic, okay, can ferment lactic acid as well as propionic acid and produce carbon dioxide. Okay, now this is lactic acid bacteria. This is where the proprinium bacteria are very important. They are found in Swiss cheese production. They are found also in the intestine and anaerobic environments of the skin. Moving on. Anoxygenic Phototrophs. These are photosynthesizing. So phototrophs, they need light. The difference is that they don't produce oxygen, as they use hydrogen sulfide or organic compounds for the source of electrons. The two major groups, purple bacteria, which is due to their light harvesting pigments. They appear purple, red, or orange. You're going to see purple sulfur bacteria. Some have a flagella. They're found in sulfur-rich habitats, accumulate sulfur granules in the cytoplasm. These include chromatium, thiospirillum, and thiodictium. The purple non-sulfur bacteria, so they don't use sulfur, use organic molecules as the electron source and reside in bogs and most soils. And they include Rhodobacter. Now, green bacteria. They're green or brownish in color, have a chlorosome. Basically, they have a light-collecting accessory pigment sites, and the two types, green sulfur, green non-sulfur. Green sulfur use hydrogen sulfide as a source of electrons, but secrete sulfur granules to extracellular medium. These include chlorobinium and pallidodicium. Green non-sulfur use organic molecules for reducing power. Okay? Now, what you're seeing there are the images of basically cyanobacterium. Okay? Don't panic, we're going to get to them. And finally, we have other anoxygenic phototrophs. Several newer species exist. Helicobacterium, which are gram-positive endospore-forming rods, and they're related to Clostridium. For the oxygenic phototrophs, okay, ancestors were created, basically their ancestors created the oxygen atmosphere. You have to think about this. 4 billion years ago, 3.5 billion years ago, we didn't have an oxygen atmosphere. Yeah, we had methane, we had some ammonia, we had lots of water, we had carbon dioxide, da-da-da. You had to push everything over to have oxygen. Why? 2 reasons. One, oxygen allows you to have aerobic metabolism, but the oxygen also helps protect the environment. Everything in an anoxic environment lived in the water. Why? Because the UV rays were not stopped because there was no ozone layer. And so they would come down onto the surface of the Earth and basically destroy everything. Remember, lots of UV is a bad thing. Well, what happened is, as oxygen levels were rising, the oxygens would start reacting with the UV light and starting to form ozone. And the ozone layer was eventually developed, which allowed for life to eventually evolve onto the land. Okay? So, what are we talking here? Basically, oxygenic phototrophs use oxygen as a terminal electron acceptor, and they use light for energy. They fix nitrogen for other organisms that are nitrogen deficient. They may contain the enzyme nitrogenase, which is an enzyme complex that mediates nitrogen fixation. Cyanobacteria form filamentous multicellular associations called trichromes, and these include Anabathia, Synchronous, and Trichodecium. Now, aerobic chemoliths. What are we talking here? Ugh, that's a yucky looking stuff. But that's when you have excessive growth of cyanobacteria. Okay? That's when you sometimes will get what they call eutrophication. There's a massive amount of fertilizer and other materials, and this will stimulate the formation of this green yuck. Some people consider it algae, and in some cases, yes, it is an algae overgrowth, but other cases, it'll be because of cyanobacteria. And if you look here, this is an Anabathia species with its heterocyst. Okay. Now, moving forward, we have aerobic chemotrophs. They obtain energy by oxidizing inorganic chemicals. Now, inorganic chemicals don't contain carbon, sulfur, nitrate, ammonia, hydrogen gas. They use oxygen as a terminal electron acceptor. Some can even oxidize iron and manganese, and there's several types. The sulfur oxidizing bacteria play a critical role in the sulfur cycle. They recycle sulfur in the environment. They use oxygen as a terminal electron acceptor, and they end up producing sort of a sulfuric acid. The filamentous sulfur oxidizers include Bigiata and Thyrothrix, the unicellular sulfur oxidizers, Thiobacillus. By the way, if you're wondering what all these things are, anything with THIO is referring to something with sulfur. Okay? They cause bioleaching, leaching of metal ions due to the conversion of insoluble metal sulfides into soluble metal ions. And they produce sulfuric acids. This is where you get sometimes some swampy situations or water that gets pumped out from mining situations, and it's very acidic. And the reason it is because the bacteria started to live off that, and one of the things that they end up producing as a byproduct is sulfuric acid. So they tend to cause the water that's being pumped out of the mines to be very, very acidic and can be very damaging to the environment. Nitrifiers are a group of gram-negative bacteria. They obtain energy by oxidizing inorganic compounds, usually inorganic nitrogen compounds, ammonium nitrite. Some convert ammonia to nitrite. They include Nitrosomas, Nitrobacter, and Nitrococcus, which converts ammonia to nitrates. Okay? You had seen this, the sulfur oxidizing bacteria. The upper one, of course, is Bigiata, and the lower one is Thyrothrix. And we're going to move from here after we get through the hydrogen oxidizing bacteria. So we had sulfur, nitrogen, hydrogen, okay? Don't get totally wrapped up in this because we will eventually get to some of the really cool stuff that causes diseases and things, so hang in there. Hydrogen oxidizing bacteria. Usually thermoflix will be Equifex and Hydrogenobacter. Now, we're going to move over to page 285. And now we're going to start getting into the aerobic chemoorganic trophs. They oxidize organic compounds for energy. They use oxygen as a terminal electron acceptor. What do we got? We got major classes. The obligate aerobes. They've got to live in oxygen. They obtain energy by respiration. Micrococcus. These are gram-positive cocci that can withstand arid and up to 7.5 salty habitat. Micrococcus luteus, which you see there, luteus meaning yellow, produce yellow color colonies. Mycobacterium produce including saprophytes, do not have a cell wall but have a waxy compound in the cell membrane, and they require acid-fast staining. Yep, folks, these include Mycobacterium tuberculosis and Mycobacterium leprae. They form what's called pleomorphic rods. And finally, we got Pseudomonas. Now, these are gram-negative rods. They produce pigments. If you look there, you're saying, wow, there's red, there's orange, there's yellow, there's green, there's blue. The different colors are because of the water-soluble pigments produced. But hey, the big thing that you know is Pseudomonas Aeruginosa, which is an opportunistic pathogen. It infects patients with underlying medical conditions. It's usually referred to as a nosocomial infection, and it's not something you want to play around with. Now, thermos. These are thermophilic. They produce the Taq polymerase, Thermus aquaticus, thermostable DNA polymerase. Deinococcus are highly radiation resistant. Okay? And what you're going to see here is we're going to talk about next the facultative anaerobes. Now, keep in mind something. If the difference between facultative and obligate. Obligate is got to have it, no negotiation. Facultative is I can live with it, I can live without it. I'm adjustable. And therefore, if you say facultative anaerobes, they prefer oxygen, but they can live via fermentation. They can get without oxygen, no problem. Corynebacterium. And we see that here. They produce metachromatic granules. They're gram-positive pleomorphic rods. They form club-shaped cell arrangements. Enterobacteria, when we talk about the intestine, they're found in the intestine. But also some of them will be found in very rich soils. They're also known as the enterics or enterobacteria. They're gram-negative rods. They include Proteus, Enterobacter, Klebsiella, Shigella, Escherichia, Salmonella, Yersinia. Beginning to sound familiar? They're going to get more. Coliforms. These are enterics that ferment lactose. The general structures can be useful for identification of strains and select species. Vibro, typically found in marine environments. Most require low levels of sodium for growth, so they've got to have some sodium present. They're gram-negative, straight or curved rods. Vibro cholera also causes the cholera disease. Vibro parahemolytic causes mild GI illness. Vibro vulnificus. These usually cause illness leading to liver disease. Now, we're going to start moving into the ecophysiology, which is the study of adaptations by organisms of select habitats. This might seem like a lot, and I am going to skip a section. If you'll note, I put it in big bold print between 290 and 294, and also skip pages 294 and 295 to the magnetobacteria. The point I'm trying to get you to understand is the disease pathogens that we're going to be dealing with in the coming weeks. What are their habitats? Where do they come from? Where do they favor? And that may relate to where they're located in the body. You got to remember that bacteria can live in a wide array of habitats. For many, the relationship of the anatomy and their physiology is due to the habitat that they live in. When we talk about terrestrial environments, endospore formers, okay, these include Bacillus and Clostridium. Okay, what you're looking at in figure 11 and 14, you see megaspores in the bacterium megatherium. In the lower picture, you have endospores forming at the ends of cells. That's Clostridium tetani. It causes tetanus. Okay? So, basically, what's so important about this? This allows these bacteria to last a long time. They can go into dormancy until habitat conditions are favorable for growth. Endospores are the most resistant type of dormancy. Now, by the way, this includes various species like Bacillus anthracis, yes, that is the pathogen that causes anthrax. Various Clostridium species, such as those that cause tetanus or botulism. Now, Azobacter forms a cyst. And you can see that here. And it's a cyst-resting cell type But this is resistant to ultraviolet light, drying, but not heat, and it results from a thickening of the cell wall. Now, by the way, going back just one, I encourage you to take a peek at the physiological diversity. These are going to help you to understand some of these, okay? Now, myxobacteria, during emergencies, they end up forming this fruiting body structure, as you see here, okay? And which helps to distribute dormant cells in the form of a spherical microcyst. This is a structure resistant to heat, drying, and radiation, but it really is less than an endospore. And then, of course, you've got Streptomyces, which encompasses about 500 species. They're gram-positive. The growth resembles a fungi, strangely enough. Okay? And you see that here. People would actually look down on that skull and say, oh, that's a fungi. No, that's not. They're gram-positive. They form branching filament mass called mycelium, and the spores are called conidia. They produce a wide array of antibiotics. So later on, when we talk about antibiotics in some cases were actually produced by certain Streptomyces species that we have been able to find will overcome certain organisms. Now, we got to move all the way over to page 294 as we start talking about bacteria with unusual mechanisms, or movement mechanisms, I should say. And the first one we come up against is spirochetes. This is a gram-negative bacteria. They have spiral shape. Moved by an axial filament. It's sets of flagella set in the periplasm between the inner and outer membrane. And they cause the cell to rotate in a corkscrew-like fashion. They can move in mud, water, even bodily fluids. Leprospira interrogens, which causes leprospiriosis, is one type. Now, when we talk about spirochetes later on, we'll be talking about other various types here. But what we're going to do, and I know that's going to be getting some people excited. I'm serious. So excited. Why? Because you took the course because you wanted to know the medically important bacteria. Well, here you are, table 11.3. Don't worry, you don't have to lose all the sleep tonight trying to memorize everything here, but you're going to get introduced to a lot of these as we move on, particularly the last third of the course. We're going to go through lots of this stuff. So what we've got to really start thinking about is the animal or the human being as a habitat. Now, bacteria that will inhabit the skin. What about the skin? Well, skin's usually dry and it's somewhat salty. And so that's where you're going to get Staphylococcus. And Staphylococcus happen to basically like this environment. They're facultative anaerobes. Staphylococcus epidermis is harmless. Staphylococcus aureus is horrible, particularly in wound infections and also in other diseases. We'll get into this. Staphylococcus saprophyticus is a urinary tract infection, a UTI. And then we end up starting to go from the skin to the mucous membranes. Now remember, the mucous membranes include respiratory, digestive, and general urinary tract, the GI tract, and the GU tract. Much of it is an anaerobic habitat. So keep in mind that we're going to be dealing with Streptococcus, which is the respiratory tract, Cornea bacterium, which is the respiratory tract, Lactobacillus, which is in the vagina. It will convert glycogen to lactic acid. Clostridium is in, of course, the intestinal tract, and Enterobacterium is the intestinal tract. Now, bacteroides are strictly anaerobic. They're gram-negative rods, and they have the coccobacilli format. Bacillus fragilis and relatives constitute 30% of the fecal bacteria. This is where we get into the microbiota that you're talking about with the intestine. Lots of bacteria in there. And some of it isn't necessarily bad, except when it gets out of the place it's supposed to be. Bacteroides can cause blood infections and abscesses after intestinal surgery or appendicitis. Bifidobacterium are anaerobes, rod-shaped, gram-positive. They ferment sugars in the intestine to produce lactic acid and acetic acid. Okay? Campylobacter are curved gram-negative rods. They're microaerophiles. Campylobacter jejuni is a diarrhea disease in humans, and it can be found in poultry. And this is usually one of those types that's a food-borne disease. Raw chicken Sometimes people would get Campylobacter if the chicken has contaminated a table, cross-contamination, which I'll mention in the food microbiology later on. Okay? And these situations, you take the raw chicken, put it on the grill, but the raw chicken plate, you take the chicken off the grill, put it back on that same raw plate where the raw was, the bacteria are there, and now you consume the chicken. That's why you have some situations of foodborne illnesses. Helicobacter, which is a curved gram-negative rods. They're genetically related to Campylobacter. They're microaerophiles. H. pylori, Helicobacter pylori, causes ulcers in the stomach and duodenum. They have the enzyme urease to neutralize the acidic habitat. Okay? Now, Haemophilus, tiny gram-negative coccobacilli. They thrive in blood. They're found in the respiratory tract. Now we're going to start getting into Haemophilus influenzae, which causes ear infections, respiratory infections, and meningitis. Haemophilus ducreyi causes sexually transmitted disease, chancroid. Okay? Now, we've moved from some of these types, and we're going to move into some other interesting ones. Now, what you're looking here is mycoplasma colonies, if you're wondering. We're going to be getting into mycoplasma in a second. And actually, I try to have some added images beyond the book to enhance your understanding. Let's get to Neisseria. Neisseria is a gram-negative kidney bean-shaped cocci, usually in paired cells. They're obligate aerobes. Many are nutritionally fastidious, meaning it's usually really hard to culture these guys. You've got to have a really, really buffed up, specific type of culture medium. Okay, so you're going to have a specialized one, and sometimes it's used with differential to just separate out all that you need from the bacteria to just get Neisseria. And Neisseria gonorrhoeae causes gonorrhea. Neisseria meningitis causes meningitis. Here are our mycoplasma. They're among the smallest forms of life. They have no cell wall. They have sterols in the cell membrane. Mycoplasma pneumoniae causes pneumonia. And you can see the sort of like fried egg appearance of the colonies you saw previously and in this one. Now, Treponina are spirochetes. They inhabit body fluids. They're gram-negative cell walls. They're obligate anaerobes or microaerophiles. Treponina pallidum causes syphilis. Borrelia is also a spirochete, but they are also microaerophiles that are transmitted by ticks or lice. Borrelia recurrentis and Borrelia hermesii causes relapsing fever. Borrelia burgdorferi, yes, transmitted by ticks, causes the Lyme disease. Now we're going to get into the obligate intracellular parasites. Look at obligate intracellular means what? They have to be inside the cells. In essence, they're unable to reproduce outside the host cell. These are bacteria that have lost the ability to produce substances necessary for their own extracellular growth, so they have to basically inhabit inside cells. And that's the only way. We're going to get into this because some of them are extremely medically important. Okay? So, all in this table are spread, and you see the table Rickettsia orienta and Ehrlichia are spread by blood-sucking arthropods, either a tick or a louse. All of them are gram-negative rods or coccobacilli. Rickettsia rickettsii causes Rocky Mountain spotted fever. Rickettsia porwatziki causes epidemic typhus. Orienta susugamushi causes scrub typhus. And if you're sitting there going, how does he know this stuff? I took a course purely on Rickettsia, which included also covering such things as Orienta, Ehrlichia, etc. And I had to learn the diseases as part of my doctorate. Ehrlichia chaffinensis causes human ehrlichiosis. Okay? They've all got to live inside the cells. The next image you see is of Coxilia. This organism is the only one that survives well outside the host. Coxilia burenti causes Q fever. So Coxilia can be transmitted without an insect vector, forms a spore-like structure. which allows it to survive outside the host. It's usually acquired by inhaling bacteria shed from infected animals. So in abattoir slaughterhouses, things like that, where they're doing also shearing of wool, etc. from sheep, it was first found, and they called it Q fever because they really didn't understand. The Q meant unknown, till they figured out the entire role of coccilia. And this was also considered at one time a bioweapon. Okay? Now, chlamydia is transmitted directly from person to person. It's gram-negative, but lacks a peptidoglycan. Inside the host cell, it forms a reticulate body that reproduces by binary fission. And you can see the images here, which are of chlamydia. It develops to an infectious form called an elementary body, and so the elementary body is passed. Okay? Now, we come up toward the end, and so what diseases do you see with chlamydia? Okay, first, chlamydia trachomatis, which causes eye infections and mimics gonorrhea, which is an STD. Chlamydia pneumoniae causes atypical pneumonia, and Chlamydia piscatiae causes psittacosis. Now, psittacosis is a form of pneumonia. It's usually related to being passed on through birds to humans. Now, we got another one which I wanted to include. It's included in the book, but I thought it would be important since it's becoming very, very contemporary in research now. Wolbachia. Now, Wolbachia pipinitis infects arthropods, insect spiders, mites, as well as parasitic worms. And you're sitting there going, big deal. I'm not going to treat insect spiders or mites. But it can be used for mosquito control. You see, when the infected female insect is basically infected with Wolbachia, any male embryos that she's carrying die. Infected male mosquitoes infect females, and thereby you limit the population. It is seriously being considered now in the control of Zika and several other mosquito-borne diseases. Now, there's specific strains of Wolbachia that will do certain things. Also, Wolbachia infects the microfilary of Anoxygenic Phototrophs. This is the parasitic worm that causes river blindness. You see, the worms are transmitted through, if I remember right, it's either a mosquito bite or a, I think it's mosquito bite. Basically what happens is these worms will start moving around. They eventually get up to the eye and lead to damage to the eye. But they found that if they treat the patient to destroy the Wolbachia bacteria with doxycycline or tetracycline, that sterilizes the worms, it stops the microfilary production, and reduces the severity of the microfilary disease. In other words, if you treat the bacteria, which are a necessary symbiote, they live inside of these microfilary, and they're destroyed, the worms are inhibited and will eventually be destroyed. So, this is why it's so important. And you'll see this mentioned in perspective 12.1. It's a little advanced in the next chapter, but it's well worth reading. Finally, we come up to a couple of examples of archaea. Now, remember that archaea thrive in extreme conditions, temperature, salinity, acidity, alkalinity. They inhabit anaerobic niches, except for the methanogens. Now, again, this is where you just have to be familiar with them, not have to memorize everything. Why? Because if you take a look at one image, this is a solar evaporation pond. You've got that red area there that's really concentrated salt. And yet that red area there is being created because of the Halobacterium and the Halorubium, etc. Okay? The other situation you have is basically the this is the sulfur hot springs in Yellowstone National Park. Typical habitat of Sulfolobus. Now, extreme temperatures and in some cases extremely acidic conditions. Now you might be saying, so what? What you have to understand is that from an industrial viewpoint, these bacteria can provide you with certain proteins that might be used in washing clothes that can handle high temperatures, like hot water. They can break down oil, grease, and other materials, and they might be used for new tools in genetic engineering or in biotechnology. So I wanted to just bring them up to you. Now, we're just about at the end, and what I would encourage you to do is take the time to review, of course, the chapter summary and prepare for next week. And thank you so much for your time. Have a great day.